All right, hello and welcome back. Today we're talking about blood. This is a very short lecture, thankfully, as opposed to some of our previous lectures, which can go quite long. Blood is one of the shortest lectures of the semester, so there's only going to be one video for blood, and then we'll be able to move on to bigger and better things. So what I have here is a picture of more or less the family tree of blood. Up here at the top, we have what's called the hematopoietic stem cell. This is just the stem cell that all other blood cells are derived from. Now, most of this big complicated stuff in the middle, you guys are not going to be responsible for, but it can be kind of useful to figure out how we actually determine the types of cells that you guys will need to be familiar with. So where do platelets come from? Where do red blood cells come from? Where do the various white cells come from? You can kind of figure out how they're related to each other through this tree. But we'll talk a little bit more about it at the end. But just to kind of calm your nerves a little bit, most of the stuff in here you don't really need to worry about. So whole blood is the stuff you just take out of a vein. You'll have many different types of material there. If you remember, blood is a type of connective tissue. It had its extracellular matrix was a liquid called plasma, and it does have cells, sort of. Erythrocytes, our red blood cells, make up about 44% of volume of blood. Though it should be pointed out that erythrocyte is not technically a cell because it doesn't actually have a nucleus. So RBC, red blood cell, erythrocyte, these are actually kind of misnomers or mistakes. Platelets are also fragments of cells. They are not true cells. And because of this condition, where we have some things that look like cells but are not actually cells, blood, all the components of blood taken together are called the formed elements. Because you really can't call them all cells because they're not cells, so we have this term formed elements. Now, one group of cells that are actually really cells are leukocytes. So leukocytes are white cells. These are true cells. They do have a true nucleus, and they're correctly named. So the three basic components of whole blood, if you take it out of somebody's arm and spin it down, the bottom 44% will be red blood cells. There's going to be this little white layer on top. It's sort of pearlescent white layer that's called the buffy coat. This very thin little layer is where you're going to find your platelets and your white cells. And sitting on top of that will be the liquid portion of blood, and that is called plasma. That makes up about 55% of the volume of blood. So all these cells taken together, all these cell fragments taken together are called the formed elements. So here is the image to make things a little bit more clear. Stuff on top, this is just liquid portion of blood. That is your plasma, 55%. The bottom 44% are all red blood cells. This little 1% in the middle is your buffy coat, which contain your platelets, which are very important for clotting of blood, and your various white cells that are going to protect you from infection. Now, the job of the red blood cells is to help transport oxygen. If we look up here in the components of the plasma, you can see that the vast majority of it is made of just simply water. There are a significant amount of proteins dissolved in here: albumins, globulins, fibrinogen. Albumins and globulins, I'm not going to talk about. I'm going to leave that for your physiology class. We will briefly mention fibrinogen. Fibrinogen is a protein that is also involved in the clotting response. And there's going to be 1% other solutes, so nutrients, electrolytes, ions, gases, things like that. So the plasma also helps deliver nutrients and gases inside blood. So just to be clear, when you take blood out of somebody's arm, you're not going to see this until you put it in a centrifuge and you separate materials by density. So the heavy red blood cells go to the bottom, white cells are on top, and then you have the liquid portion on top of that, just in case that wasn't clear. Let's move on. Functions of blood, super important for transportation of oxygen. So we need to get oxygen from the outside into our blood so our blood can carry it to our cells, so our cells can carry out aerobic cellular respiration. Pardon me, a little frog in my throat. So our cells need oxygen in order to generate energy, and that comes ultimately from the outside. When our cells burn fuel to make energy, they're going to produce the waste product, carbon dioxide. So carbon dioxide is a toxic compound, and it needs to be removed from our cells. And that is also a major function of blood. It will transport oxygen to cells and then pick up the waste carbon dioxide, transport it to the lungs, and then we're going to exhale that carbon dioxide. Blood will also carry other nutrients, sugars, things like that, what the cells need to survive. And it will also carry away other waste products, things like urea, for example. Blood is also important for help regulating body temperature. So I think we mentioned this way back in the very first day of class. Something that can happen if it gets very cold outside, blood vessels are going to close off inside your skin, and that will direct blood away from the skin towards deeper structures in your body. That will help you conserve heat, so you're less likely to lose heat. Conversely, if you're very hot, like when you're exercising, blood vessels on the surface of your skin are going to open up. Blood is going to go to the surface of your skin, and it's going to make it easier to radiate off heat. Blood is very important for regulating body pH. The regulation of blood pH is very, very important to the functioning of blood. You can only have about 2/10 or more than 2/10 of a change in blood pH can lead to very serious health effects. It can actually be lethal. Go more than 2/10 in one direction or the other, a little bit more acid, a little more basic, you're either going to go into coma or you're going to go into seizures. So blood pH must be very, very tightly regulated. And that's something you'll go into a lot of detail when you get into physiology. But here, I'm just telling you it's important. Fluid levels, right? So all fluids of the body are derived from blood. Tears, cerebral spinal fluid, milk, right? All these things are derived from blood. Protection. So blood, you will have these white cells. White cells are going to travel around our body in our circulatory system, and they can exit our circulatory system and deal with any problem like an infection we have in our body. Another thing that you find inside blood are called antibodies. So an antibody is an immune protein that can help fight infection and fight pathogens. Now, this table covers lots of useful information, but I'm not going to go into extreme detail here. But this is very useful for you when you guys go on to physiology. So I am going to present it, but I'm not going to test you on it. So it will talk about what are the function of albumins. It's important for maintaining osmotic pressure, and what are the kinds of things that globulins do. Go ahead and read this, because while your head is in the game of blood, this will help lay the foundation for physio, but I consider this information to be physio and not really anatomy, so I'm not going to really test you on it, but it's useful to know. Right. So there's another term we need to describe here, and that is hematocrit. So a hematocrit is really a count. It's the number of the different types of cells and structures that you have in blood. How many red blood cells do you have? How many of the different type of white cells do you have? How many platelets do you have? Now, this number and the ratio of these numbers can vary depending on age and gender. You might be asking yourself, why do we care? Well, knowing what an individual has and comparing that to a standard can give you an idea of something, or what might be wrong with a patient. If you find a patient that has 15% fewer red blood cells than they should have, right, that may be an indication that the person has anemia, right? They're going to have a hard time transporting oxygen. They're going to be tired all the time. It's going to be very hard for them to do a whole lot of anything. It'd be hard for them to get out of bed. And you might go looking for the reason, well, why do they have anemia? And see if they have a blood parasite or something like that. So it can be an indication of disease. So typically what happens, somebody doesn't feel well, and it isn't like immediately obvious, like they don't have like a knife sticking out of their head. The doctor looks at somebody and they're like, I don't feel good. They're like, run a blood test. Let's see what's wrong with them. So bits of information down here that are useful. So this column, and that column, I do want you to be aware of these. So I do want you to know that an erythrocyte is about 8 micrometers in diameter. That's an important landmark. I do want you to know that they're very important for transporting oxygen and carbon dioxide. And I do want you to know that they're going to last about 3 or 4 months. So your average red blood cell is only going to last 120 days. Then it gets broken down and recycled. The exact number, maybe you have to know that in physio. I'm not going to ask you that level of detail in this class. Leukocytes are white cells. They are substantially larger, or some of them are larger than red blood cells. So they're anywhere from 1 to 3 times the size of red blood cells. So it helps make them identifiable. What they do, they're going to help protect us from waste, cellular debris, and protect us from infection. Their lifespan can vary quite dramatically, so don't worry about it for right now. If you take an immunology class, then you can get into it. Platelets, these are very small. These are tiny little fragments of cells. What is their job? They're involved in help blood clotting, right? So if we didn't have any way of clotting blood, even a very small hole in our circulatory system, everything would just bleed out, right? That would not be good. So clotting is just a way to plug those holes so we don't bleed out, or exsanguinate is the technical term for it. These don't last very long, so in the order of a little bit more than a week. And you don't need to memorize the exact number of how many platelets your average person has. This is just a slide telling you how to prepare a blood smear. I'm not going to test you on this, but realize that if you were just to take blood out of an individual, put it on a microscope and take a look at it, you would see only 2 kinds of cells. You would see red blood cells because they are red, because they're filled with hemoglobin, and you would see clear colorless cells. These are the white blood cells. Now in this image here, the white cells are blue and purple because a chemical has been added to it, a stain that will turn nuclei a dark color. Notice that the red blood cells are unaffected because they don't have a nucleus. They don't have a nucleus, the purple stain won't have any effect. So when you look at a stain like this, if it's a Wright stain or something like that, Gemsa stain, you can see that a chemical has been added because the white cells, you can see the nucleus, you know a chemical has been added. So the steps of how exactly you do this, don't worry about it. But I just want you to realize when you look at a slide like that, you see these purple cells. The purple cells are really white cells because if you didn't stain it, they wouldn't have any color. They'd be white. Okay. I think I've made that clear. All right, specifics of erythrocytes, also known as red blood cells. As I mentioned, these are not true cells because they don't have a nucleus and they don't have organelles. They're about 8 micrometers in diameter and they have a biconcave shape, right? So concave means it makes a little dip. So the cell kind of looks like that. I'll have a picture in a second. The reason for this shape is to maximize the volume to surface area ratio. Because remember, what's the job of a red blood cell? It's to bind oxygen and then release it. So you want to have a very large surface area on these cells to maximize the ability to release oxygen and absorb oxygen when needed. If you have a line of red blood cells all piled up in a single file, it's called a ROLU. Most capillaries, the really small blood vessels of the body, they're only about as wide as a red blood cell. So you get all the red blood cells all lined up, and that structure is called a ROLU. So there it is, ROLU. So the space is a capillary, that's a single red blood cell, a bunch of them all lined up in single file is a ROLU. So here's the shape of a red blood cell. Notice that it has that concave shape. Why does it have this shape? To make it easier to give off oxygen when it's in the various tissues in the body and to be able to absorb oxygen quickly when it's in the lungs. So the actual compound inside blood that allows for the binding of oxygen is a protein called hemoglobin. There are about 280 to 300 million hemoglobin molecules inside a single red blood cell. So the book quotes 280 million hemoglobins per red blood cell. So this is capable of binding and releasing oxygen depending on the environmental conditions. So I'm not going to go into that. That is a topic for physiology. Just realize that hemoglobin can bind up to 4 oxygen, and it can release them under different conditions. The hemoglobin molecule is what is called a heterotetramer. So tetramer means 4, so it has 4 basic parts. 2 of them are identical. So you have what are called 2 alpha subunits and 2 beta subunits. That's what is shown here for 2 alpha chains, 2 beta chains. Each one of those chains is called a globin. So if we go to the picture, this whole thing here is the hemoglobin molecule. One of these is 1 alpha chain. Here is our other alpha chain. This is 1 beta chain. This is the other beta chain. The reason it's called heterotetramer, hetero means different, because you have 2 alpha and 2 beta. Tetramer means you have 4 pieces, 1, 2, 3, 4. Now, each of these globin molecules, each of these chains has a heme group, which is this little guy here, which looks like a little planet Saturn. If you take a close-up image of that, you'll see this structure. This is heme. So you see this complicated organic molecule. Don't worry about the details of that. What you have in the middle is an iron atom. So this is the structure that allows for the binding of oxygen. So oxygen binds at the iron in the heme. Each hemoglobin has 4 heme groups, 1 in each of these domains of hemoglobin. So again, we don't need to worry about the specifics of this. Right now, I'm introducing the structure to you. How this works. Why does it bind oxygen in the lungs and release oxygen out in the tissue? That is a subject for physiology. Okay, so I believe I mentioned this, but just to go over these slides, right? A heme group contains that organic molecule. This thing here is called a crown ether. If that doesn't mean anything to you, don't worry about it. In the middle of it, you have an iron molecule. Here is where oxygen is going to bind in hemoglobin. Let's look at the life cycle of a red blood cell. Red blood cells are created inside bone. You might remember that from our bone chapter. So red blood cells are created inside bone, and then they're going to migrate into our circulatory system. There, they're going to run throughout our body, delivering oxygen, carrying away waste products, all that good stuff for about 3 or 4 months. After that time, the cells get kind of beat up, they get kind of old, and they're going to be absorbed and recycled by the body. So it's the job of the liver and the spleen to clean up the old, dead red blood cells. So old cells are going to be phagocytosed or gobbled up inside the liver and spleen. The cells are going to be broken down, and special mechanisms are in place to take care of the heme groups and the iron from the hemoglobin. Now, I'll get back to this in a second. The iron and heme are going to be separately processed by the liver, and the remaining components are going to go back to the bone in order to make more red blood cells. So we just have this recycling pattern going on. So let's get back to the heme and the iron. Why is that relevant? Why am I making a special purpose of this? Well, iron is very tightly controlled inside the body. You have almost no free iron floating around your body. Your body works very hard to make sure this stuff is either inside a red blood cell or it is inside special transport proteins that doesn't allow this stuff to float around free. And the reason that is bacteria really want that iron. They want that iron bad because it allows them to make special biochemical pathways that allow them to produce a lot of energy. So your body does not want unwanted bacteria growing inside of it, so it tries real hard to keep all this iron bottled up. Also, it's convenient because it can then be recycled and sent back to the bone marrow. So One thing to remember, no free iron in the body is a way to suppress bacterial infection. Now, over here, the heme. What is the story with heme? Well, heme gets broken down in the liver and it forms a substance called bilirubin. Now, you may or may not be familiar with bilirubin, but bilirubin is a breakdown product of heme. If bilirubin builds up and your body is not able to further break down the bilirubin, you become jaundice, right? So your bilirubin has a yellow color. So people who have liver damage, they get cirrhosis, they get a condition called jaundice. Their skin turns yellow. I mean, yellow, like their eyes turn yellow. And the reason is that they can't get rid of the bilirubin that's building up in their system because their liver's not functioning properly. And this stuff can be really, really toxic. So why do people turn yellow? Because their liver stops working, because the liver can no longer process the bilirubin. So the way the process works, heme is collected from old red blood cells. Heme is turned into bilirubin. If the liver is functioning properly, bilirubin is then broken down into bile, right? And so bile is how bilirubin is going to leave the body. Bile will actually be used by the liver to help digest food and as a way to get rid of old heme. So there's really kind of like a double function, a double whammy there. So just a little fun fact, why people who have liver damage due to cancer or infection like hepatitis or because of cirrhosis from drinking too much, they're going to turn yellow because their liver can't process the bilirubin and their skin's going to change color. Also, there's another time when people check bilirubin levels at birth. So anyone who's had a child, one of the things, what they call, I think, like an APGAR test, one of the tests they do on the newborn is they do a bilirubin test, because oftentimes when we're first born, our liver is not yet fully functional. And the process of birth leads to the rupture of a lot of red blood cells, and you may have a buildup of bilirubin. So it's just a way to monitor the newborn to see whether or not their liver is functioning and if they need any extra support. Okay, so our next topic, we're going to talk about different types of blood types. You guys have probably heard, I'm A positive, or I'm B positive, or I'm O negative. So we're now going to talk about what exactly is going on there. So it says here, erythrocytes, red blood cells, contain surface antigens. Now, for those of you who don't know what an antigen is, repeat after me. An antigen is a compound that provokes an immune response. An antigen is a compound that provokes an immune response, something that makes your immune system do something. Is this good or is this bad? Well, it depends. If a bacteria is causing your immune system to become activated, that is a good thing, right? You want bacteria to be antigens. You want your immune system to react and attack unwanted bacteria. There are situations where you can have a self-antigen. So a self-antigen would be an example of one of your body's proteins makes your immune system attack, right? That would be bad. That's an example of an allergic reaction or an autoimmune response. So what rheumatoid arthritis is, right? Anyone seen their mother or grandmother have rheumatoid arthritis? All the hands get kind of gnarled and the fingers get twisted. What's happening there is your own immune system starts attacking the tissues in your joints and causing all that damage. So that's your body's immune system attacking you. So self-antigens, bad. That's an allergic reaction. That's an autoimmune response. You do want your body's immune system attacking foreign invaders. So just remember that an antigen is a compound that provokes an immune response. So all of our blood cells have these antigens on their surface. It helps identify. Now, in comparison to other tissues of the body, blood is very, very simple. The system of antigens is very simple. If you just think about it for a second, right? It's really easy to transfer blood from one individual to the other, right? It's just a simple ABO system. anyone who's ever known anyone who needed an organ transplant, that is really hard. You want to take an organ out of somebody and put it to somebody else. That's like a 1 in a million situation, right? It is very difficult to find a match for organs. And that's because most of the other tissues of the body have many other markers. They have many different types of markers. Blood is very simple. We only have these few. That's why it's relatively easy to exchange blood between different individuals, but very hard to exchange organ tissue between different people. Okay, that aside, let's get back to our blood types. We're going to have antigens, just signaling molecules on the outside of our red blood cells. We have three basic types. The first is going to be the A antigen, and then the B antigen. These are simply sugars. These are sugars on the outside of our red blood cells, which our immune system may respond to. Blood type O simply means you do not have the A sugar or the B sugar. So it basically is nothing. And I'll have an image here that should make this clear in a second. In addition to these antigens, in addition to these sugars in our red blood cells, blood will contain certain kinds of antibodies. So an antibody is an immune protein that can bind antigen. So if for whatever reason you have the A antigen, the A sugar on your red blood cell, and you have an anti-A antibody, meaning an antibody that binds the A antigen, that binds the A sugar, those two are going to get together and you're going to have an immune response, right? We don't want that. You don't want your immune system attacking your own red blood cells. So the way we understand this terminology, an anti-B antibody, an antibody is an immune protein. What does it recognize? What does it attack? It attacks the B antigen, the B sugar. If you have an A antigen and an anti-A antibody, they're going to bind together and it can lead to things like agglutination, which basically means globbing together. Your blood is going to turn to oatmeal. You don't want your blood turning to oatmeal. That's why you have to be really careful when giving somebody a transfusion. You give them a compatible blood type. So you don't have this agglutination, you don't have hemolysis going on. Let's cut to the picture. It should be a lot easier. So here we have the various blood types. Here we have the red blood cells and the signal molecules, the antigens on them. And here are the different types of antibodies that each blood type will have. So blood type A means you only have the A sugar. You only have the A antigen present. Now, those people are alive, which means their oatmeal is not blood, which means they do not have the A antibody. Because if these guys had the A antibody would attack their red blood cells and their oatmeal would turn to blood and they would die. That doesn't happen. So if you're blood type A, you have the anti-A antibody, but you don't have the anti-A antibody, but you do have the anti-B antibody. And likewise, if you're blood type B, you will have the B sugar on your surface, but you will not have the A sugar, which means that you will have the anti-A antibody, but you won't have the anti-B antibody. Otherwise, the immune system attack your blood cells and you would never be born. Now, if you're both blood type A and B, your blood type AB, you're going to have both the A sugar and the B sugar on your red blood cells, which means you have neither antibody. Because if you had either anti-A antibody or anti-B antibody, it would attack your blood and then it'd be all over. So as I mentioned, blood type O means you don't have an antigen. You don't have the A sugar, you don't have the B sugar. In that case, you're going to have both antibodies. So the way to remember this, I know it's a lot of terminology as I'm trying to work through it. Blood type A has the A sugar. Blood type B has the B sugar. Blood type AB has both sugars. Blood type O has neither sugar. Now, the way the antibodies work, During fetal development, self-reactive antibodies are destroyed. So in utero, very early on in development, when your immune system is being created, you may have a cell that's going to produce an antibody that will attack your own red blood cells. Your developmental process will destroy those antibodies. They will be removed. Otherwise, you would never basically make it out of your mother's womb. So any self-reactive antibodies are suppressed and removed. If you've heard of this process before, it's called clonal deletion. If you haven't heard of that, fine. So blood type A, you got the A sugar, you'll have the anti-B antibody. Blood type B, you're going to have the B sugar, which means you're going to have the anti-A antibody because the B antibody was removed. Blood type AB, you're not going to have either antibody. Blood type O, you're going to have both. Now, the reason this is relevant is because when you put all this together, you can start to understand who is the universal donor and who is the universal recipient. And you'll go over that probably in more physio, but there's one other point I want to talk about before we get to that. And that is the plus minus thing. So you've heard of O minus, O negative, or O positive. Well, plus and minus refers to simply a different sugar. In this case, it's called the rhesus factor. So rhesus factor, you either have the rhesus factor or you don't have the rhesus factor. So if you have the rhesus factor, you're positive, Rh positive. If you don't, you're Rh negative. So in this case, we just have a different sugar shown in blue. So if you're Rh positive, right, trying to limit, if you're Rh positive, you're not going to have the antibodies. If you're Rh negative, you may or may not, and I don't want to open up that can of worms because it gets confusing. So, to more fully understand the compatibility of donors, there's something you need to keep in mind. When you donate blood, right, they stick a needle in your arm, they drain blood out of your arm. Then what they're going to do is they're going to put it in a centrifuge and spin it down. So when they put it in a centrifuge, they're going to separate the liquid component from the cellular component. All of your antibodies are in that liquid component. So when you donate blood, you're only donating red blood cells. You're not going to donate the antibodies because they're going to separate that away. So that is why type O is the universal donor, because their red blood cells do not have any markers on it. So when you take O blood cells and put them in another individual, that individual has antibodies in their blood. But even if they have both the A and B antibodies, those antibodies have nothing to bind to. So to be more specific, the true universal donor is O negative because they don't have the A sugar, they don't have the B sugar, they don't have the Rh factor, therefore no antibodies bind to them, so you're not going to have any trouble. So that is why type O negative is the universal donor. So do you want to be type O negative? No, you want all your friends to be O negative because they can donate to you. What you want to be AB positive. If you are AB positive, that means you have the A sugar, you have the B sugar, and you have the Rh factor. If you have all those factors, you have none of the antibodies, which means you are the universal recipient. So what you want is for you to be AB positive, but all your friends be O negative. That way you're in the best possible position. So something to keep in mind. And then from here, if you keep in mind who has what sugar and the recipient has certain antibodies, you can work out how all these work together. I'm not going to grind through the whole process. I'll save that for physio. But just so you understand the basic structures in the ABO system and how it works, and you can sit there and figure it out if you need to. Now, this shows the agglutination reaction. These are one of the two most common problems you can have if you have a blood incompatibility. Oops, you injected the wrong blood into the patient. You gave them a blood cell that had an antigen that is recognized by one of their antibodies. Now, lots of things can happen when an antibody binds an antigen, but two of the big ones are the antibody will cause that cell to be destroyed. So you just put in fresh red blood cells and they all get blown up. So it's not going to have any effect, right? That's not good. You're also going to cause inflammation. Another thing that can happen is what's shown down here. You give the wrong blood type, the patient's antibodies start forming a network or a chain of red blood cells, and that leads to a blood clot inside the blood vessels, right? That leads to your blood turning to oatmeal, right? We don't want that happening because all the capillaries that service things like your lungs and your brain and the rest of your tissue aren't going to function, and that's going to be a real bad day for everyone involved. So make sure you know your blood type compatibilities and don't make any mistakes because you might get an agglutination reaction. All right, next up are leukocytes, or the white cells. These are the cells that are going to help protect our body from bacterial infection. They are true cells. They do have a nucleus. They do have organelles. They tend to be a little bit larger than red blood cells, and they're capable of these two very fancy words. Let's take this one first, chemotaxis. Chemotaxis. Chemo means chemical. Taxis, you guys remember, anyone here old enough to remember long time ago, you used to like dial this number on a phone that had a cord, and then this yellow car would show up outside your house. That was a taxi, right? It's a taxi is a place that takes you somewhere. So chemotaxis literally means movement based on chemicals. So leukocytes can smell chemicals in your blood, and they can travel from point A to point B. So the motion of cells directed by chemical signals is called chemotaxis. That is how cells get from where they are, how the leukocytes get from where they are to where they need to be to defend you from an infection. Diapedesis, this is a fancy sounding word. What diapedesis means is when a cell that's circulating around in your vessels leaves the blood vessel and enters the surrounding tissue. So what these guys are going to do, if this is the wall of your blood vessel, these white cells can wiggle their way out and exit your circulatory system and enter your tissue. So how do white cells travel around your body? In your cardiovascular system. How do they know where they need to go? How do they know where the infection is? They follow their nose. They do chemotaxis. Once they get to a particular region of your body where they're seeing these signals, they leave the circulatory system through diapedesis and then start fighting the infection. All right, classifications of leukocytes. We have two basic classes of white cells. We have what are called granulocytes and agranulocytes. Granulocytes have visible granules. A means not. So agranulocytes are cells that do not have visible granules. So the three kinds of granulocytes are neutrophils, eosinophils, and basophils. Do you need to remember that? Yes. Neutrophils, eosinophils, and basophils are all granulocytes, which are all leukocytes. Agranulocytes include lymphocytes and monocytes. Be careful. Do not confuse lymphocytes with leukocytes. All lymphocytes are leukocytes. Not all leukocytes are lymphocytes. Lymphocyte is a subclass of leukocyte. The words sound kind of similar, and people tend to get a little bit confused by that, so don't fall into that trap. Okay, so lots of good information on the slide. Put stars across the top. I would expect you to be able to recognize these different types of cells if I give you a blood smear image. So I might give you just a slide of blood, and I might ask you to tell me what all these cells are. Easiest one, right? Red blood cell. Okay, that's easy. These are all red blood cells. These top three are our granulocytes because you can see really obvious big granules, right? Big blue granules, kind of pinkish granules or orange granules over here, and big purple granules in this one. These do not have large visible granules. They have kind of a grit to them, but they don't have big granules, these little spheres. So our agranulocytes, our lymphocyte, and our monocyte. Let's go through these one by one and learn the characteristics by which we can identify them. So first up is the neutrophil. The neutrophil is the most common type of leukocyte we have. So you look under here, neutrophil, 50 to 70% of all leukocytes, 50 to 70% of all white cells are neutrophils. So when you look at a blood smear, you're going to see a lot of these. They have dark blue granules, and they have a multi-lobe nucleus. So they may have 4 or 5 lobes or like little chunks to the nucleus. It looks like sometimes like a string of sausage. That is one of the ways you can identify a neutrophil. Very common, dark blue granules, multi-lobe nucleus. Very important for fighting bacterial infection. Now over here, we have what's called an eosinophil. Eosinophils are a little bit bigger than neutrophils, right? You can see that it's bigger compared to the red blood cells than the neutrophil. It has a bilobe nucleus, so it has 2 lobes to the nucleus. Right? 1 lobe here, 1 lobe there. Be careful, right? You're not necessarily always going to see both lobes of the nucleus. If the eosinophil is rotated in a certain way, you're only going to see 1. So you have to get lucky to see it in a way where you can see both halves of the nucleus. Because you turn it that way, you're not going to see both lobes. Only here you would. So bilobe nucleus, and we're going to have orange or pink granules. That is how you're able to identify eosinophils. Eosinophils are relatively rare. Look down here. Only 1 to 3% of leukocytes are eosinophils. These guys are important for fighting parasitic infections, particularly large things like worms and flukes. Now over here, we have a basophil. Not that you can tell from this picture, but basophils also have a bilobe nucleus, a nucleus with 2 halves. You can't see it here because there's so many granules, they're blocked. Oops. Here, the granules are dark purple. So bilobe nucleus, dark purple granules, you know it's basophil. These are by far the most rare. 1/2 to 1% of all leukocytes are basophils. So these guys are really rare. It's pretty hard to find them. The job of the basophil is to release inflammatory compounds. What am I talking about? Excuse me, histamine. So when you take an antihistamine, you have an allergy, you take an antihistamine, you're trying to prevent cells like this one from releasing histamine, which causes your nose to run and your eyes to water and all that kind of stuff. Okay, so that wraps up our granulocytes. Neutrophils, eosinophils, basophils. Neutrophils, a little bit bigger than red blood cells. They have dark blue granules and they have a multi-lobe nucleus. Important for fighting bacteria. Eosinophils, a little bit larger, they have a bilobe nucleus and they have pink or orange granules, important for fighting worms. Basophils, bilobe nucleus, and they will have dark purple granules and they release histamine. Do you need to know all that level of detail? Yes, and it is right here. Now our agranulocytes, we have a lymphocyte. Lymphocytes are fairly easy to identify because they're about the same size as red blood cells, but they have an enormous nucleus. In fact, this one has a relatively small nucleus. Oftentimes, you won't even see cytoplasm. You'll just see the nuclear membrane and then the cytoplasmic membrane right on top of it. You won't even see the difference between the two. So an enormous nucleus, that tells you it is a lymphocyte. What are lymphocytes? These are, in case you've heard these terms, B cells, T cells, natural killer cells. These are all examples of lymphocytes. This is part of your adaptive immune system. If none of those terms make sense to you, don't worry about it. Lymphocytes make your B cells and your T cells. Over here, we have what's called a monocyte. Monocytes are really big. They're like 3 times the size of a red blood cell, no granules, and their nucleus looks like a kidney bean, usually. These guys are fairly rare, only about 10% of cells in your blood. And I should point out that the next most common type of leukocyte are your lymphocytes. So lymphocytes, B cells, T cells, quite common. Monocytes, fairly rare, only about 10%, really, really big kidney bean shaped nucleus. Right. What these guys are going to do is they're going to exit your blood supply and help fight infection in other parts of your body. So by listing the cells by which are most common, neutrophils are the most common, 50 to 70%. Lymphocytes are the next most common, 30 to 40%. After that, I would say would be monocytes at around 5 to 10%. Eosinophils down by like 3%, and basophils are the most rare at 1% or less. So you might be saying to yourself, well, why do we care about this? Well, remember, I talked about hematocrit, knowing the various differences between the cell counts. So you don't know why somebody feels bad. You take a blood sample and you have the person down in the lab count the cells. If the person has way more neutrophils than they normally should, that may be an indication of bacterial infection somewhere in the body. You can't see it in the surface. They have a bacterial infection. So you can start looking for bacterial infection. What if they have like 10 or 15% eosinophils? That would be a way overproduction of eosinophils. It probably means they have like a worm infection. They may have a bunch of worms in their intestines, or maybe they have lung flukes and their body is trying to fight it. So by seeing that you have elevated eosinophils relative to neutrophils, that gives you some idea what's going on. Somebody has really bad allergies, they may have an overproduction of basophils, right? So you can use this information to make clinical diagnoses. All right, so that's it for white cells. Now let's start talking about platelets. These are the small little fractions of cells that help clot our blood. Platelets are literally fragments. They are little pieces broken off of bigger cells. And those big cells are called megakaryocytes, right? So that's not, you know, from some monster movie. A mega just means big. Karyo means karyon. This means nucleus. So these are cells with really enormous nucleus. That tells you exactly what it looks like. Here we go. A megakaryocyte. Where are these guys located? Inside red bone marrow. They're sitting out here in the red bone marrow, and they have these little tendrils, these little pseudopods, which extend into the capillaries in the bone marrow, and just little bits of it break off, and that's how platelets are created. So they're fragments off these megakaryocytes, and they play an important role in having our blood solidify and prevent us from bleeding out. So what actually makes a blood clot is not just platelets, but it also involves red blood cells and another protein that I mentioned before called fibrin. I think before I called it fibrinogen, so that was a component in plasma. Whenever you see gen at the end of something, pepsinogen, fibrinogen, that means it's a precursor. That means it has not yet been activated. Fibrinogen is dissolved inside your blood and is constantly circulating. When the clotting cascade is activated, fibrinogen turns into fibrin. Fibrin makes this big tangled rat nest of protein. So fibrin mixed with platelets and red blood cells is what actually makes a blood clot, and that's what's actually going to plug the hole in our circulatory system. Now, the process by which blood clots are created is very complex. There's this long chain, there's this long domino effect of various proteins. Your physio instructor may or may not mention it, but it's something like a 30-step biochemical pathway. It's pretty awful. We're not going to talk about any of that here. Okay, I think the last topic we're going to talk about is hematopoiesis or hemopoiesis, and this is just the process by which we generate the formed elements, or the cells of the blood. So we're going to start with a pluripotent stem cell. That just means that it's a stem cell that can turn into any of the different types of blood cells. And that cell is called a hemocytoblast. So we have one cell that can turn into any of the red blood cells, any of the white cells, any of the platelets, any of that stuff. And that's called a hemocytoblast. The hemocytoblast will take one of two pathways. It can either go down the myeloid line, or it can go down the lymphoid line. I'll have a picture of this in a second. And it's pretty easy to tell them apart. Here's our hemocytoblast. If it goes down the lymphoid line, it will become a lymphocyte. That's it, lymphocyte. I mentioned before, we have 3 different types of lymphocytes: B cells, T cells, and natural killer cells. Everything else, all these other cells, are derived from the myeloid line. So the hemocytoblast can turn to the myeloid line. The myeloid line might turn into a red blood cell, or it might turn into a megakaryocyte, which then creates platelets, or it might turn into a cell that will eventually create a eosinophil, basophil, neutrophil, or monocyte. Again, all this stuff in the middle, you don't need to know the particulars of unless I lectured on them. You need to know what a megakaryocyte is, right? The P thyroblast, no, you don't need to worry about any of that kind of stuff. So if I didn't lecture on it in any detail, you can just forget about it. I'm just showing you the family tree for the different types of red blood cells. So that brings us back to the figure that we started with. Here's our hemocytoblast. It can then turn into the myeloid line, the lymphoid line, where you're going to get B cells, T cells, or natural killer cells. Everything else goes down the myeloid line to get platelets, red blood cells, or our granulocytes, or agranulocyte. This table is kind of useful. The reason why I included it is it tells you the locations of where these cells are found. Everything here in this yellow, all this area here, these are all precursor cells. That stuff all happens in the red bone marrow. What do you find in blood? That's this layer right here: platelets, red blood cells, basophils, eosinophils, neutrophils, monocytes, natural killer cells. What do you actually find in the tissue? The cells that have exited the circulatory system and are now inside our tissues. You have things like mast cells, dendritic cells, macrophages, right? We talked about some of these cells way back in tissue. So there were certain resident cells that help protect us. Examples like dendritic cells and macrophages. So again, what is the important takeaway in this slide? This middle section here. What are the cells you actually find in blood? The rest of the stuff is just sort of gee whiz information for you. And with that, we've come to the end of our section on blood. So that went a little bit longer than I had anticipated. Hopefully that is fairly clear to you. If not, I will be available online for office hours soon. Till next time, take care, stay safe, stay healthy. See you soon.